I would like to introduce Dr. Dom Gallery. Dr. Gallery joined Mote, located in Sarasota, Florida in 2024 as a postdoctoral fellow in the Seagrass Ecosystem Restoration Research Program after completing a PhD in ecology, evolution, and behavior at the University of Texas at Austin. Their graduate research evaluated the differences between cryptid lineages of two Caribbean coral species using genome-wide scale approaches. So through a combination of field and lab research, they focused on complex interactions between corals' genetics and environments to assist with conservation managers and restoration efforts. Now, as a postdoctoral fellow at Mote, Dr. Gallery uses their expertise in genomics to identify seagrass genotypes with resilience to present and future environmental stressors. Similar to Mote's successful coral restoration program, they will build and maintain a genetic library of seagrasses to be used in restoration efforts with collaborations from all around the state of Florida. And with that, please join me in welcoming Dr. Gallery.
Thank you. All right, let me share my screen. Okay, so thank you so much for that introduction. Today I'm going to be talking to you about the genetic diversity of shoalgrass or Halodule wrightii across the Gulf Coast of Florida. But before we dive in, I want to make a couple of key points. So first, seagrass meadows, like coral reefs, are a vital marine ecosystem with the potential to sequester almost 20% of the world's oceanic carbon. They're a food source for many animals, such as turtles and manatees, provide habitat for juvenile fishes and invertebrates, and improve water quality by trapping sediment and absorbing nutrients. And seagrass meadows are threatened by changing seas, so they're vulnerable to stressors such as pollution, climate change, habitat destruction. And according to a journal article that was published in 2009 using a meta-analysis of available seagrass research, we are losing approximately 7% of seagrass beds globally. And so it's of utmost importance to work in earnest to recover the seagrass ecosystem, hybridizing restoration techniques with cutting-edge technologies to reestablish the seagrass ecosystem, especially here in Florida. That being said, one of the reasons it's so important to restore seagrass is they are one of the least protected marine ecosystems. According to the United Nations, seagrass beds are among the least protected of marine ecosystems at only approximately 26% protected versus 40% of coral reefs and 43% of mangroves. So today I'm going to talk to you about what we can do, what I've been doing with my research in seagrass genomics. But before we dive into how we can assist restoring seagrass, let's take a quick step back and talk about what are seagrasses. So for those of you who may not be aware, they are a true plant found living in saltwater, which unlike land plants have had to evolve some unique characteristics. So as halophytes, they are able to regulate their osmotic pressure by moving salt ions into their cell walls and vacuoles. And like land plants, they have rhizome and fleshy roots that anchor them to the sediments in the sea floor. And to reproduce, they release pollen and gelatinous clumps that are carried by water currents to flowers where they produce seeds. And in Florida, we have 6 native species of seagrass that form the seagrass ecosystem. So we have widgeon grass, shoal grass, turtle grass, manatee grass, stargrass, and paddlegrass. And they are truly important ecosystems. They provide support for wildlife, they provide economic support, and they're ecologically important. So for wildlife, they provide ecosystem services, habitat, nursery, and food sources. They provide shelter for many species, including juvenile fish, invertebrates, algae, and bacteria. It's a food source for many animals, including fish, turtles, manatees, octopuses, shrimp, oysters, and clams. And they're biodiversity hotspots that support many species. Economically, they support humans for recreation, water quality, wave attenuation, and natural heritage sites. They provide nurseries for commercially important fish species, which help support local economies. They're also early indicators of ecological health, because they are sensitive to stressors and contaminants, it can indicate the ecological health of coastal areas. And seagrasses also trap soil and sand with their roots, which helps prevent coastlines from erosion. They're also great at water quality, so they filter water by trapping sediments and absorbing nutrients and pathogens. They also produce oxygen through photosynthesis. They store carbon dioxide in their leaves and roots and can store carbon up to 30 times faster than many forests. They also protect vulnerable ecosystems and species, such as coral reefs, by acting as an acidification buffer. So real briefly, we're going to talk about what is threatening seagrass. So I highlighted here 6 different things that are threatening seagrass. It's not an all-inclusive list, but it is some of the top stressors that we are studying. So the first one is salinity. So hypersalinity, which is too much salt, can be caused by droughts and dry seasons, and hyposalinity, which is too little salt, can be caused by storm runoff and wet seasons. There's also increased temperatures that seagrass have to contend with. So increased water temperatures and severity and frequency of heat waves, prolonged heat waves, have increased the amount of time that bottom water temperatures are higher than normal. And this can be detrimental to the seagrass ecosystem. There's also eutrophication, or nutrients in the water, which it can be caused by high phosphorus inputs, which it can increase algal blooms, which outcompete the seagrass. It can also increase epiphyte growth. So epiphytes are small diatoms and algae that live on the seagrass leaves that can block the seagrass from getting light. Speaking of talking about blocking light, water clarity or turbidity is also a very important stressor threatening seagrass. So this can be caused by sediments from rivers or the bottom of the ocean, dredging, flood plumes. And this increase of suspended sediments or chlorophyll A or dissolved organic matter blocks the light that seagrass need to photosynthesize. The next stressor for seagrass is hypoxia or anoxia. So a reduction or complete loss of oxygen in the water. And high biological oxygen demands from increased water temperatures can lead to sulfide toxicity from increased anaerobic bacteria. And finally, herbivory. So when there are too many herbivores in the water from poor management practices, we can have biomass reduction from overgrazing, which can decrease seagrass productivity and alter species assemblages. Now, all of these things are threatening seagrasses in Florida. And like I said, this isn't an all-inclusive list, but it is a pretty expensive one. And we want to identify how to assist seagrasses in Florida through restoration efforts. But first, we need to know what seagrasses we have or what genetic diversity of seagrasses we currently have. So I mentioned that there are 6 native species of seagrass in Florida. However, my research focuses on just one of them, Halodule wrightii, or the shoal grass. So Halodule wrightii is a tropical to subtropical species of seagrass found in the Western Atlantic, in Florida, the Gulf of Mexico, the Caribbean, Central and South America, and the Eastern Pacific. And it's a pioneer species, so it's one of the first seagrasses to colonize disturbed habitats, typically found in shallow, high light, high temperature, and variable salinity environments. And it relies on a combination of genetic and clonal strategies to persist in these unstable environments. So Halodule wrightii reproduce both sexually with flowers and seeds and asexually with rhizome extension. Many meadows are dominated by clones that can persist for decades, and sexual reproduction may be episodic and environmentally triggered, for example, by temperature stress or disturbance. And so what this implies is there's high clonal dominance within sites, which causes low genotypic diversity within a meadow, but sexual recruitment events can rapidly increase genetic diversity when they occur. So Halodule also has adaptations to survive environmental stressors. So like I said before, it tolerates high temperatures, wide salinity ranges, high light, high turbidity. And it does this with genetic variation, which increases its thermal tolerance, linked to fast growth rates and high stress recovery. So as a genetic model species for seagrass, Halodule wrightii are a great option because they have a wide geographic range, they have strong environmental gradients, they have a mix of clonal persistence and sexual reproduction, and they are used frequently in restoration because they are fast to establish in disturbed regions. So they are a strong candidate for studying different factors such as local adaptation, resilience genes, and climate change responses. And they're ecologically important, but genetically understudied compared to other species such as Thalassia. So patterns of genetic diversity in Halodule wrightii. The genetic diversity varies within meadows, among meadows, and across regions. Genetic structure often shows isolation by distance, and clonal growth can stabilize local meadows. In previous microsatellite studies commonly find low genotypic diversity locally, for example, clone-dominated patches, moderate to high allelic diversity regionally. Genetic studies also suggest connectivity among populations, especially along coastlines, and long-distance dispersal maintains gene flow and evolutionary potential. And so Halodule wrightii maintains genetic diversity at regional scales, even when local meadows appear genetically uniform. And so all of these are great reasons to study Halodule wrightii. So they show clonal growth, variable genetic diversity, and tolerance to environmental stress. But the gaps do still remain. So what I'm studying specifically is, does this genetic diversity relate to thermal stress tolerance? So talking more about my research projects themselves, we are looking at genotype responses to thermal stress. So to do this, we collected 90 unique genets from 3 estuarine regions along the Gulf of Mexico: Sarasota Bay, Charlotte Harbor, and Estero Bay. And then once collected, each genet was split into 3 sections, and the apical meristem was removed if possible. Each section had at least 1 shoot, 1 root, and 1.5 centimeters of rhizome. And we measured physical parameters such as the number of shoots, the length of the rhizome, and the number of roots at the beginning of the study. We also collected tissue samples for genetics, transcriptomics, and microbiome studies. And so each section of a genet was placed into a treatment tank, one of 3 treatment tanks. Each genet had an individual clone in the control, the mid-temperature and the high-temperature treatments. The control temperature is current high day temperatures in Sarasota Bay at 28 degrees Celsius. The mid-temperature experiment is the predicted end century temperature if there is between 2 to 4.5 degrees Celsius warming. And the high temperature is if the predicted end century warming is between 5 and 8.5 degrees centigrade. We then allowed the seagrass to acclimate after being cut and placed into these pots for approximately 2 weeks. We then ramped up the temperature slowly in the treatment tanks over 8 days. We held those seagrass at the maximum temperature for 2 weeks, ramped down over 8 days. And now they are in the recovery phase currently, which has lasted about 4 weeks. It ends on Friday. And so at each of these time points, we measured the leaf growth as well as take a small tissue sample for transcriptomics and microbiome. And what we expect to see is that there is genotypic variation between sites. So what we see there is we expect to see 3 subpopulations that are strongly correlated to the location where the seagrass came from. And we expect to see a correlation between that and their heat tolerance. Therefore, the southernmost sites, for example, Estero Bay, would outperform fitness measures for Charlotte Harbor and Sarasota. And so gene expression and microbiome will also correlate between losers and winners, as we call them, so those that grow faster versus those that grow slower. And we are hoping to potentially identify genes and or alleles that convey more heat stress tolerance so that we can use these for selecting individuals for restoration purposes. And so the collected data here will inform what genotypes are resilient to thermal stressors. And once we've determined which genotypes should be used in restoration, we need to develop a plan for restoring these genotypes to nature. And here I want to pivot to put this research into context of the larger goal at Mote and across the state of Florida. So we have the Seagrass Restoration Technology Development Initiative, which was signed into law in 2023. And the law states, in collaboration with the program DEP, Mote Marine Laboratory, and the University of Florida shall create a 10-year Florida seagrass restoration plan to implement tools and technologies developed under the initiative. Basically what this looks like is we have a series of different plans and administrative reporting goals, such as 6-month technical and financial reports, competitive calls for research proposals, annual reports, a technical advisory council that advises the seagrass teams on what research should be performed. Then we are also developing a 10-year seagrass restoration plan and hosting education and outreach workshops. And the initiative is also in charge of leveraging funds to provide research equipment and operations. So we did build a genetics lab and greenhouse nursery here at Mote Marine Laboratory in Sarasota. And we also provide staffing, equipment, supplies, and maintenance for folks coming to do research here. We also have field operations where we travel for field seagrass and stressor research. And so the Seagrass Research Infrastructure provides multi-user seagrass research infrastructure for initiative scientists, which is free of charge for all initiative projects. The Seagrass Education and Outreach is also available for folks who are studying seagrass. And we test a variety of field stressors here at the greenhouse in Sarasota, including temperature, light, salinity, nutrients, and pH in a controlled setting. And this can assist with determination of resilient seagrass genotypes, so those individuals that are more likely to outperform under stressful environmental conditions. And this is really important because restoration is both expensive and time-consuming, and you do not want to spend a lot of time and effort outplanting seagrass that are going to immediately die because of stressful conditions in the field. And so this is just a quick schematic of what our greenhouse currently looks like. We are in the process of expanding the greenhouse and building another building as well for looking at carbon sequestration of seagrass. So the first 2 years of the initiative focused on examining the genetic diversity of seagrass populations and associated field stressors across Florida, and developing a genetic library of Florida seagrasses. So how this works is folks do great research. They send us samples. We process and genotype all of their samples and provide them with that information, as well as house that information in perpetuity so it can be used for restoration goals in the future. We are also testing the resilience of different statewide seagrass populations to multiple environmental stressors in a controlled lab and nursery setting. We are examining existing seagrass restoration technologies and conducting planning efforts to pursue regulatory approval for testing novel seagrass restoration technologies. And then finally, we're organizing and encouraging collaboration among the scientists and restoration practitioners working on Florida seagrass genetics to achieve the goals of the initiative. And so we have several accomplishments and priorities, including developing a great team of scientific partners and staff, which we are always looking to expand. Every year we put out a call for proposals. And then we also use leverage funding to develop a genetics lab and purchase the equipment needed to do the genetics. And so the management plan review is just the last step of this seagrass initiative. And this is where we reviewed over 116 documents looking at the different reports from aquatic preserves across the state, as well as estuary programs, state parks, and looking and seeing what has been done previously on seagrass and what needs to be done moving forward. And so we have a 46% loss of seagrass since 2014 within the boundaries of the estuary programs. And 26 plans that we reviewed included seagrass restoration planning, and 7 of those had plans with seagrass restoration targets. And 3 plans actually had large targets with projected restoration greater than 2 acres. However, there is no statewide seagrass restoration plan currently, and there are no plans that propose using genetically resilient seagrass management activities. That being said, there are 2 plans that acknowledge the needs for genetically resilient seagrass, so the Pensacola and Perdido Bay Estuary Program and the Florida Keys National Marine Sanctuary Revised Management Plan. And so the framework to building a 10-year seagrass plan involves a lot of reading papers and reports, as well as identifying and cataloging relative seagrass restoration activities and capturing all these lessons learned. And we hope to put all of this in together with the University of Florida and Florida Department of Environmental Protection to create this 10-year resilient seagrass restoration plan. So with that, I'm happy to take any questions about either the Seagrass Initiative at Mote and UF, or about my research.
Awesome. Thank you so much, Dom. That was super interesting and awesome to know that we've got a big seagrass team working towards getting us restored from those seagrass losses. So yeah, thank you, everybody. You'll see a poll if you guys could please fill that out when you're able to. But for now, if anybody has any questions, please feel to type them into the chat, or if you want to unmute and ask Dom directly, now would be a great time. And I can moderate the chat here. So we have a question from Stacy Cecil, and they said, "Are you interested in exploring additional stressors like light stress or low salinity?"
Yes, absolutely. So the project I just am in the process of completing right now on thermal stress was a pilot project. So it was a proof of concept that we could actually split the seagrass in this manner, grow them up under stressful conditions, and get good genomics and transcriptomic results. So we absolutely plan to expand to other stressors such as light stress, low salinity, pH. Yeah.
Awesome. I had a question for you. So, I know you mentioned that like you've got some samples in the work here, and you're moving forward with some of this genetic work. What are you kind of expecting or predicting in terms of the genetics of who's going to be winners and who's going to be losers in terms of patterns?
Yeah, absolutely. So I think there will be some level of where you are from that will help predict your level of success in in the genetic patterns. So, an individual from a region in Estero Bay that experiences very high temperatures continuously every single summer is likely to outperform an individual from Pensacola that is more used to maybe cold stress. I do think there will probably likely be trade-offs though between stressors. So when we start looking at some different experiments where we're doing compared stressors, we'll probably likely see that maybe you do better in thermal stress, but not as well under salinity stress, which I think will very much contribute and be important for us during restoration because we'll want to outplant a wide range of genotypic diversity when we're doing restoration efforts.
Yeah, awesome. So it sounds like you're gonna have to pick the winner that's best for the situation because no
Yeah.
one seagrass is gonna be perfect.
Yeah, I'm not a strong believer of the ideal individual model.
Yeah, okay, got it.
Yeah.
Awesome. I have another question here from Smitty, who's asked, are you doing any analyses of gene expression? If so, what are the genes of interest you're looking at?
Yeah, that's a great question. So we are doing transcriptomics data. So the approaches that I use are transcriptomic wide, so genome wide. I don't target a specific gene, so I'm not looking for a specific heat stress gene or salinity, um, salt ion transfer gene. I'm looking at the genome or the transcriptome overall to see if there is clustering of individuals and groups of genes. That being said, we're hoping that we will identify some genes of potential interest that we might be able to then start looking more at doing like qPCR techniques to look for those specific genes or alleles in seagrass instead of having to undergo an entire stress test for every seagrass genotype because there's a lot of them.
Oh, cool. And for those of the people that might not know, what is a QPC? I think you
Oh,
said.
oh, a qPCR.
Yeah,
It's a quantitative PCR. So it's a technique where you take a very small section of the gene and you just amplify that section of the gene to see if it's being expressed or not in the individual. So you're looking for just a specific gene.
got it. Okay,
Cool.
thank you.
Yeah.
Not a genetics person, as you can tell. I have another question from Julie Christian. Have you collected halodule from other areas of Florida as well?
Yes, absolutely. So we have Halodule samples from collaborators from Pensacola. We have samples from Dr. Reynolds from the Nature Coast and the Big Bend region, as well as Indian River Lagoon. And we have some Halodule from the Florida Keys from Dr. Robert Johnson.
Awesome. Um, Another question again from Stacy Cecil. Are you or any of your lab mates investigating genes related to hormone expression related specifically to growth and rhizome robusticity under stress situations?
Not currently, but that is a great avenue to expand this research into. So yeah, I really appreciate you noting that. That is a great place to sort of look into in the future.
Awesome. All right. Does anybody else have other questions for Dom? Oh, and I see the poll just came up. All right. Well, I had one more question for you, Dom,
Yeah.
and this is more of like a philosophical question, but I guess to you, what would if you could have unlimited restrictions, what would be an ideal seagrass restoration project and site, and how would that look like for you?
Yeah, for me, that would be a site where we're able to restore multiple species of seagrass with a lot of genetic variation within those individuals that we're restoring that also have these genes that are, or these alleles that are resilient to the different stressors specific to the region that um, seagrass restoration is happening. And then they would all live forever.
Yes, and they would all live forever, happily ever after.
Yeah.
Great. All right. Well, thank you everybody for coming. Any last questions before we sign out here? All right. Well, thank you so much, Dom, for joining us. That was really awesome. And you guys can catch us all in the next Nature Coast Currents in a few weeks. But until then, take care, everybody.